and I'll move us into the last section of our day one presentations. So next, I'm very excited to introduce Dr. Alba Grifoni to the virtual stage to present using the IEDB in your research. Thanks very much, Alba.
Thank you so much, Nina. Hello, everyone. As you might heard, I'm a little bit under the weather. However, I hope I can still convey how excited I am about IEDB. I've been using IEDB in my research since over 12 years, even before joining the La Jolla Institute. And when I joined, I had the pleasure also to meet them in person, and that was a fantastic experience. And so I hope my presentation is gonna be able to show you what can be done with IEDB to answer some interesting scientific questions. And so I am going to focus today mostly in coronaviruses because many of you have experienced the pandemic, and probably many of you are still working in understanding what's happening to our immune system the more our T cells or immune system in general is exposed to different variants. And so at the beginning of this year, we actually were wondering regarding a specific SARS-CoV-2 variant. The name given by the media was Pyrola. The scientific name is BA286, and essentially the reason why this variant in particular was raising concern is that because this specific variant had over 60 different amino acid mutations within the spike protein from the original SARS-CoV-2 that we got in 2020. And the difference between this BA286 and BA2, which was one of the circulating variants, was pretty much 30 amino acid mutation, which is what we used to have when the original Omicron actually hit. And so a little bit the question was, how is our immunity wall built from previous infection or vaccination is gonna able to overcome or recognize BA286? And so to answer this question, we leverage information available in the IEDB. So we extract SARS-CoV-2 epitopes using one of the queries that Randy has shown you before. Additionally, we apply a filter for canonical size, and that was because we were interested really in focusing on epitopes for which we knew there were restrictions, and I'm going to show you in the next slide why. And then once we extract all the different epitopes, we went into ImmunomeBrowser to really make sure we were looking at ancestral epitopes. And the rationale is that IEDB is a database that keeps evolving through time. And many studies now have been focusing on the different variants. So in our case, we really wanted to understand if we go back to epitopes that are identified when you got the first COVID vaccine, and then we compare them with the most recent variant, what is the effect of this mutation in these original epitopes? And so here is a summary of the epitopes that pertain the different SARS-CoV-2 proteins. And then in the pie chart below, I show you what fraction of these epitopes are actually 100% conserved, meaning there is no absolute mutation. And then roughly 28% of these epitopes actually contain a BA286 mutation. In diving more, the majority only contain a single mutation. So one of the questions that we wanted to really understand is how impactful are these mutations in the epitopes that we see in the IEDB? And so to answer that question, we went back to IEDB again. And this time, when we look for the epitopes, we also extract the different restrictions. So the idea here is, how can I combine information that we have in the database, so experimental data, with applying prediction to see how a situation can evolve? And so this is what this specific query was about. Once we get the epitope restriction combination based on experimental data, then we went back and used the IEDB tools to really ask, if I look at my original ancestral epitope, and then I look at the peptide that contains the mutation for BA286 in the same combination, so with the same restriction, do I still have a peptide that is going to be able to bound to that HLA or not? And so this is a recap of our analysis. Each of these data points represent a specific epitopes. On the x-axis, you have values that pertain the prediction based on the ancestral. And on the y-axis, the prediction that we derive if we look at the mutated peptides for BA286. So anything that is in the oblique line essentially suggests that there is no change in the binding ability of the HLA molecules, even if you have the mutation. Anything that goes outside is actually bad news or good news. Given the percentile cutoff of binding prediction, essentially what we see is that only 3% of our original mutated epitopes, which once again are just a small fraction, they show a decrease consistent with the assumption that these mutated peptides might not be able to be presented to the immune system any longer. So this is what I'm showing you looking at the entire proteome. So assuming that someone get exposed to SARS-CoV-2 infection, what happened if we just focus on the spike protein? And so this is the same data that I showed you before. And here below, you see the exact same data when we focus only on spike. And this is with the assumption of people that got exposed to the vaccination, but not yet to the infection. As we know right now, the majority of the worldwide population got a SARS-CoV-2 infection, which suggests that it's good news because the majority of our epitopes that are induced following infection are actually conserved, even in the most recent variant. But even in the context of spike, still 56% are totally conserved. And out of the 44 that are mutated, the majority are a single point mutation, which in a nutshell are still able to be bound to the HLA molecules and potentially presented to the immune system. So this is what's happening for the CD4 T-cell responses, but we were also interested in doing exactly the same type of analysis in the context of the CD8. So now I'm showing you the data. And in the context of the CD8, when we look at the entire proteome, the majority of the epitopes were actually 100% conserved, and only a small fraction show mutation. And most of these mutations were single point mutation. Now, as many of you may be aware, there is an intrinsic difference in the HLA class 1 versus class 2 molecules in terms of the capability to bind peptides. So in that case, even a single amino point mutation, if located in anchor position, can actually disrupt the binding. And in fact, when we look at our prediction analysis, the fraction of peptides that actually lose binding capability is much larger. When we look at the spike, we have a reduction in the 100% conserved epitopes. Once again, most of the mutations are single point mutation, and those have a consistent higher disrupt of HLA binding. So overall, even with the most recent variants, you have mutation, but the majority of your epitopes are still there. And even if you have single point mutation, particularly in the context of the CD4 T cells, we still have the capability from the HLA molecules to present the peptide to the immune system. How does it look if we look way back in the pandemic? And so here I'm showing you a bioinformatic analysis that was carried out exactly in the same way at the time where Omicron just hit us. And at the time, the original Omicron sublineages show that 88% of the response was still 100% conserved. So what we see now with the most recent variant is that this number is slowly decreasing, yet the mutations are not sufficient really to disrupt the binding, which is good news. And now I'm going to show you a final question, which is, okay, we talk about the epitopes per se, but how are those located? And so in order to answer this question, I'm going to go back to the IEDB and show you how you can query the database to really understand where are the spike epitopes located, specifically if you want to look throughout the protein. And so we love T cells, at least I do, but B cells are cool too, and IEDB actually has both. So you can select the T cell assay. You're gonna look for SARS-CoV-2. I'm gonna focus on class 2 for now, so mostly CD4 epitopes, and select the human as a host. And so you can query the database, which I'm gonna do, but I actually already did that. So these are the results that you're gonna obtain if you don't remember your query, and usually I do a lot of them at the same time, and luckily IEDB is smart and remind me what I did. So here you can see, once again, I use the organism SARS-CoV-2, and I'm focusing on class 2. So this is the amount of epitopes that we retrieve in the context of spike for SARS-CoV-2, which have actually been the result of 68 references that were curated. When we focus on the antigen, what the IDB is doing is compiling the different epitopes to the specific antigen pertaining SARS-CoV-2. And if we click in this icon right here, we can actually map and look at the epitopes in the protein. And so this is what I'm having right here. The IDB is mapping the different epitopes to this UniProt specific sequence. And here in the first upper graph that you see is the response frequency, and in particular is plotting the 95% confidence interval, lower and upper bound. So this essentially tells you what is the distribution of the data. The more the lower and the upper bound are close to each other, the more the response is actually defined. And this also suggests you have a lot of data for the spike protein. If you focus in other proteins, sometimes the range is wider, and that suggests that maybe additional information are needed, which sometimes help you to understand a gap of knowledge that you can assess with your own research. So here, as you can see, there are some specific hotspots of immunodominance in the protein that are characterized by our different epitopes. We can also look at the assay counts. So how many times did you test a portion of the specific protein and find it to be an epitope or a non-epitope? And so the assay counts allowed you to have this information. And as you can see here, we have the entire protein has been tested and find negative or positive, which kind of suggests, once again, we have a wealth, tons of data in the context of spikes, so we have really a comprehensive assessment. In other cases, you might find that the positive responses are only in specific region, and they may suggest
Alba,
maybe, yes.
Sorry for interrupting. I think your slides are stuck.
Oh, I'm so sorry about that. I apologize sincerely.
Well, no worries, but okay. Now we see the ImmunomeBrowser.
Okay, so I apologize for that. I thought I was transitioning. So let me go back to it. I apologize to everyone. Here was the original query with the epitopes that I was showing you. Here in the antigen, you can click on the ImmunomeBrowser. And this is the ImmunomeBrowser I was alluded to. So this is the response frequency, the lower and upper bound. So here you can see the spot of immunodominance. And right here, you have the epitope assay counts. As I was mentioning, in the context of spike, you have information for the entire protein, and you really can see this peak of positive responses. Right below, you have the information of each of the epitope sequence, the epitope ID that can give you back additional information around show you, and the sequence identity, which particularly in the case of the study that we did before, it was very useful for us to determine whether the epitopes was originally identified based on the SARS-CoV-2 ancestral or pertaining to one of the variants. And then here you have the number of subjects that have been tested and responded. And here is the response frequency that contains the division plus the 95% confidence interval. One cool feature that IDB has been implemented in the context of SARS-CoV-2 is the fact that we can also map specific SARS-CoV-2 variants and look at how this mutation map within the protein based on the immunodominance. And so what I'm going to show you here and what you can see is the mutation for the BA2 variant, and that was like the variant that BA286 originated from. And so now we can really map the mutation in the different immunodominant region. You have the majority of the mutation in the RBD domain, and then you do have some specific mutation. As you can see already, some of the mutation may hit hotspot of recognition, but there are a lot of spots that are very well recognized by the immune system that are absolutely untouched by SARS-CoV-2 mutation. And so this is for the BA2 variant. So what happened if we look at the same thing in the context of BA286? So I learned my lesson. I'm going to share again my screen and go back to my presentation. And so this is another way, just to recap. You can perform your query. You can look into the antigen and click on the ImmunomeBrowser, and then you can map the epitopes within the protein. And these are the assay counts. So what happened now if we look at BA286? So This is essentially a graph representing data that have been extracted from the IEDB. And so as you can see, once again, the majority of the mutations are pertaining to the RBD domain. Some set of mutation hits some immunogenic spots in the NT domain and S2 domain, but there is a lot of good regions that are recognized by the immune system that are completely untouched by SARS-CoV-2 variant mutation. Now, one point that I would like to clarify when we think about mutation, if you have an epitope and it looks exactly as it is, you're gonna bound to the HLA molecules and it's gonna be recognized by the T cells with the TCR recognition, and that's what gives you the response. When you have a mutation, you can look at different cases. So far, what we look into is mutation that disrupt the binding. Nevertheless, if you have a mutation and the binding is preserved, there are multiple situations that can occur. The first one is the TCR is preserved. So essentially, your T cells are still able to recognize the peptide even if it has the mutation. Another possibility is loss of TCR recognition. So even if the peptide is still bound to the HLA molecules, T cells might not be able to recognize it any longer. There is a third feature that we and others have been recently shown, which is the fact that the T cells are able to induce de novo recognition. Now, for some of you that have been more familiar with the SARS-CoV-2 field in general, there has been a lot of discussion about imprinting. If you receive a specific vaccine or a specific variant, your immune system may not be able to induce variant-specific responses. So our recent studies and the literature in general suggest that that is not the case. And actually, T cells that get exposed to a novel variant are able to induce specific variant epitopes following the infection. So with this novel concept, we decide to go a step further in our bioinformatic analysis and ask what happens if I simulate exposure with different SARS-CoV-2 variants, and then I compare the capability to recognize novel upcoming variant. And so in specific, what we did was, this is once again the same comparison I showed you before, looking at ancestral versus BA286. And then what we did was we simulate exposure with BA2, and then we're comparing again with BA286. So in this case, the analysis is completely in silico. So we look at the SARS-CoV-2 spike sequence, we extract the ancestral one, the BA2 and the BA286, and then we run prediction using the most frequent class 1 and class 2 alleles to ensure population coverage. And this selection has been done in the past and validated by IEDB in several publications, and is going to be object of additional information tomorrow. So the cool thing that our results show is that if you look at the comparison between ancestral and BA286, the fraction of epitopes that are 100% conserved is very low, or at least is 55% and 64%. When we move into comparing BA2 and BA286, the fraction of 100% conserved epitopes increase, essentially suggesting that getting exposed to novel variants induce the novel response that are able to catch up with the following variants. So your T cells are always there and trying to run after the virus in a way and trying to recognize it and limit the spreading of disease severity. So in conclusion, what we saw with our bioinformatics study is that non-spike regions are really less affected by mutation. And so incorporating in future vaccine candidate, not only the spike, but also other proteins might be a way to induce a more stable T-cell response through time. And the second point is really when we think about novel variant epitopes and this de novo, this is a mechanism that T cells can use in order to bridge gap the difference between the original ancestral and the upcoming circulating virus. So good news, T cells are still there, and even if the virus keep evolving, they're gonna be able to help in slowing down the spreading of the virus and reduce disease severity. Now, another question that once again we use the coronavirus example was how much in general animal models reflect human T-cell immunogenicity. So in this context, we leverage again the information from IEDB, And in these cases, we perform a query comparing SARS-CoV-2 epitopes that have been identified in the human species versus SARS-CoV-2 epitopes that have been identified in the context of mice. And in particular, we focus on H2B, and we also focus in transgenic mice. Then what we did was we leveraged ImmunomeBrowser to really combine this information and essentially provide a heat map of what regions are recognized, whether you look at the human set or the mice set. And so this is the results that we observe. Now on the left side, we're looking at HLA class 2, and on the right side, you're looking at HLA class 1. So the human HLA class 2 are here in gray and show you the refined mapping that is very similar to the data I showed you before. And then we focus on the MXC class 2, H2B in the context of mice, and we map again the overlay. So as you can see, the mice has more defined immunogenic region, some of them overlapping with the human data and some hadn't. Overall, there is a weak correlation when you compare the entire system, which is significant. Then we ask ourselves what happens if we focus on HLA that have been used in the context of transgenic mice. And what we see is that depending on the HLA alleles, you may have a better overlap or not. In general, when you use transgenic mice, your correlation increases. In the context of class 1, similarly, you do have a weak correlation between the human and the mice data, which improves in the context of specific HLA. But one point I would like to notice, for instance, when we look at the B702 data, the correlation is actually weaker than what you see in the context of mice. So it really depends what type of transgenic mice you want to use really to answer specific questions. And here is even more in depth when you want to look at the region. Let's say that you are interested in focusing just on a fraction of the protein. Sometimes if you focus only on a fraction of the protein, that might be immunogenic in the context of humans, but not necessarily mice. And so this is just something to remind ourselves how if we consider generally the ability of the human population to recognize specific response on the protein, that is much wider, and we need to be careful in selecting specific immunogenic region when we use mice model without accounting for that component. Otherwise, we miss out some important regions that are relevant in human and not necessarily in the context of mice. A final question that we were wondering is how much the pattern of recognition of class 1 and class 2 overlay. And what we saw was very interesting, as we in general see a weak but good significant correlation between class 1 and class 2 molecules, essentially suggesting that when you look at heat spot, and particularly if you focus on larger fragments of the protein, you might be able to capture regions that are relevant both for CD4 and CD8 T cell responses. Finally, all the data that I showed you so far were focusing on SARS-CoV-2, and so we asked what happened if we look more broadly to different viral indications. And so we went back to the IEDB, and in this case, we expanded our query to several viral infections for which enough information were available to really perform mapping in specific protein. And so here, as I show you, the general correlation when you compare all human data, specifically comparing with A2 and B7, which are the most frequent transgenic mice in H2B, what you can see is that although there is still a weak positive correlation, in general, A2 transgenic seems to have a better performance in representing the human data. In the context of class 2, that applies the same, with DR1 probably having a slightly better performance. So in conclusion, we do see a weak correlation between mouse responses, which suggests that H2B mice and in general, mice model can be used to evaluate immunogen destined for humans. But given that this is not happening as often, it's very important to factorize the component of what part of a specific immunogen are potentially recognized in human, and not use necessarily animal models to make a selection of what fraction are the most relevant. HLA transgenic mice have shown to have a better correlation and maybe can be used to evaluate images designed for humans, but in general, the mouse strain and the HLA transgenic has to be selected to match what is the specific immunogen region that you're interested in testing. Finally, one point that we thought it was very relevant is that some regions are actually targeted by both CD4 and CD8 T cell response. That concept goes a little bit outside the idea of actually targeting the CD4 and CD8 T cell response independently, and actually suggests that combining the two approaches to have regions that are actually immunogenic of both could be the right one to apply in future viral system. And as I said, while we use SARS-CoV-2 now in general as a model, the findings that we show are really in general applicable to different viral systems. And so with that one, I just want to spend a few minutes to thank my team. The majority of the bioinformatic analysis that I show you today were done by Tertulliano Alves, my postdoc, and also with the help and collaboration of Alessandro Sette and John Sidney. And with that one, I leave the floor to Alex and hope to answer more questions in the Q&A. Thank you.